how hormones are transported in the blood depends on their water solubility. Hormones that are water soluble, such as the catecholamines and most peptides, are simply transported dissolved within the plasma. However, poorly water soluble hormones, such as steroid and thyroid hormones, require special transporter proteins, and there are very low concentrations of these hormones present in the plasma, and this concentration is in equilibrium with the bound hormone that's present on these special transporter proteins. So what we notice is that the thyroid hormone is a good example of that. And so 99% of thyroid hormone is bound to transporter proteins. Only 1% is free in the plasma, as we see here on the bottom of our slide. And of the bound protein, 70% is bound to thyroid binding globulin, 15 to 20% is bound to albumin, and then 10 to 15% is bound to transthyretin, another thyroid hormone transporter protein. So the free hormone, in other words, this 1% in the case of plasma, in the case of thyroid hormone, that 1% is the biologically relevant hormone because that's the hormone that is available to bind to receptors. What we notice is this, that hormone concentration depends on two things: the rate at which the hormone is produced and the rate at which the hormone is removed from the blood. Removal from the blood primarily depends on excretion or metabolic transformation. This metabolic transformation will occur primarily in the liver or the kidneys. So the persistence of hormones within the plasma varies widely. Catecholamines and peptide hormones are processed quite rapidly, and so their persistence would expect to last for just a few minutes to a few hours, whereas in the case of protein-bound hormones, such as the thyroid hormones and the steroid hormones that we talked about just a moment ago, they last a longer period of time, and so several hours to even a few days is a possibility. So we see a nice little diagram from our text illustrating the eventual destination of hormones. An endocrine cell secretes the hormones, they circulate in the blood, they're excreted in the urine or feces, they're activated by metabolism, or another possibility is that they're activated by metabolism. Thyroid hormone is a good example of that. And then they bind to receptors that are found on target cells. It's also, of course, even more likely that the hormone that's circulating in the blood is actually active and will bind to receptors on target cells. Although in some cases, such as thyroid hormone, activation is necessary to increase dramatically the affinity of the hormone for the receptor. What we notice is that by definition, a hormone is a molecule that is released into the blood and circulates through the body. This then means that all cells, with rare exception in the body, are coming in contact with the hormone, and so whether a cell responds to the hormone or not depends on the presence of receptors. And of course, we're familiar with the idea of receptors. Receptors are much like enzyme and substrate interactions, a lock and key sort of fit, so that only certain hormones bind to certain receptors, and of course there are in some cases exceptions to that. But for the most part, that holds up. Particular receptors bind to particular hormones. The location, as we've said several times now, of the receptor varies depending on the water solubility of the hormone. If it's a water-soluble hormone, such as a protein hormone, the receptor needs to be in the plasma membrane, such as growth hormone. If it's a lipid-soluble hormone, such as one of our steroids, most of those receptors are found in the nucleus, simply because the hormone can pass freely across that plasma membrane. We also notice this, that the response of a cell to a hormone depends largely on the regulation of those receptors. The more receptors you have, the greater response that a cell will have to a particular hormone. It's very common for cells to upregulate and downregulate receptors in order to modulate their response to a hormone. We also notice that sometimes the same or another hormone will have an impact on the number of receptors that a cell possesses. Estrogen is a good example of this for the same hormone. And so estrogen, interestingly, upregulates the estrogen receptors that are found within a particular cell. In the case of another hormone, interestingly, what we notice is this. Thyroid hormone is a good example of that. So let's look at our little illustration from our text. If we go over here to the right-hand side of our slide, we see that when thyroid hormone is exposed to this cell, we have little or no fatty acid released. When epinephrine is exposed to this cell, we have only a small amount of fatty acids that are released from this adipose cell. But when we first expose the cell to epinephrine and then first to thyroid hormone and then to epinephrine, we have large amounts of fatty acids that are released. So in this case, we say that thyroid hormone is having a permissive effect on epinephrine. In other words, thyroid hormone is upregulating the number of epinephrine receptors that are present, so when epinephrine is released, it has a much larger impact than it would if thyroid hormone is never exposed to a cell, as we can see here. So in this particular graph, we have the amount of fatty acids released on the y-axis, and then we have the purple line illustrating thyroid hormone only, the orange line illustrating epinephrine only, but you can see the synergistic effect that occurs when the cell is exposed to both epinephrine and thyroid hormone. Of course, it has to be exposed to thyroid hormone first, and that is this so-called permissive effect, when one hormone upregulates the receptors of a different hormone. In this section, we also talk about mechanisms of hormone action. And what we notice in this case is that peptides and catecholamines can have two types of effects. One possibility is a rapid effect, and so this effect is an effect that utilizes an enzyme, a change in enzyme activity, or a second messenger. It's not what's depicted on the screen in the lower right-hand corner here. These effects are non-genomic. They don't involve transcription translation. Interestingly, the same hormone classes, peptides and catecholamines, can also have slower effects that do involve gene transcription. These effects can occur as a result of the same hormone in the same cell, but they're completely different, and they occur at certainly different speeds as a result of the mechanisms that bring them about. In the case of steroid and thyroid hormone, typically these are hormones that bring about much slower effects. They're typically transcription modifiers, as we see depicted in this little slide in the lower right-hand corner. Estrogen enters the cell, binds to a cytosolic estrogen receptor. That bound receptor serves as a transcription factor, bringing about the production of vitellogenin in this case, which is an egg yolk precursor protein, and eventually that's transported to the ovary. But the take-home message is that it's a transcription modifier, and so this is not going to happen within a matter of seconds or minutes, or maybe even hours. It's more something that's going to take hours or days to occur as a result of the time it takes for these proteins to be synthesized. We also want to mention in this particular point that hormones can also have what we refer to as pharmacological effects. Pharmacological effects are impacts of the hormone that are brought about when the hormone is present in very high levels, typically levels that are greater than would ever be observed during the course of normal physiology. A good example would be the use of cortisol and synthetic cortisol analogs to suppress the immune system and alleviate inflammation, for example. And so these are levels of cortisol that would not typically be observed in the body under normal circumstances. It is true that cortisol has very slight suppressive impacts on the immune system under normal circumstances, but those would not be sufficient to alleviate inflammation. The higher levels that are given to alleviate inflammation would be considered pharmacological levels, so levels that are not typically observed during the course of normal physiology. What we notice is that there are multiple factors that can control hormone release. What we see is that 3 of those are listed on this particular slide. One possibility are changes in plasma concentration of mineral ions or nutrients found within the body. Another possibility are neurotransmitters, and a third possibility that's fairly common are other hormones. All of these 3 are responsible in different aspects of our physiology for controlling hormone release. Now, if we go back up to the first bullet on this slide, we notice that hormones are released from the cell in one of 2 ways that we have actually mentioned before in our discussions. Either via exocytosis, like in the case of growth hormone, a large water-soluble hormone that can be maintained within vesicles within the cell, On the other hand, the other possibility for release is simple diffusion through the plasma membrane, like we see in the case of steroid hormones. Now we want to talk about briefly each of these three types of mechanisms that regulate the release of hormones in the body. The first one, of course, are concentrations of ions or minerals that are found within the body. And hormones regulated in this manner typically form negative feedback loops that control plasma concentrations of these ions or nutrients. Insulin is a perfect example. So in this illustration from our text, we notice that when plasma glucose concentration goes up, then the insulin secreting cells of the pancreas begin to produce insulin, plasma insulin concentrations go up, and that increased concentration of insulin in the plasma causes the increased uptake of glucose from the extracellular fluid to the intracellular fluid, and that means that glucose levels in the blood drop as a result of that, and so that completes our little negative feedback loop. Parathyroid hormone is a good example of the control of a hormone by an extracellular ion or nutrient as well. And so in this case, what we see is that we have a drop in calcium that causes the release of parathyroid hormone. Parathyroid hormone, via mechanisms we'll talk about a bit later, raises calcium levels back to the appropriate level. Next, we have hormones that are controlled by neurons. And so a good example of this would be the adrenal medulla. And so if we look down at our figure from our text at the far left-hand side, we see hormones that come from, we see neurons that proceed from the brain all the way down to the adrenal medulla, and those hormones control the release of epinephrine and to a lesser extent norepinephrine from the adrenal medulla. The adrenal medulla is a modified postganglionic sympathetic neuron. So it's a modified sympathetic ganglion, cluster of neurons. So there are no axons that proceed from it as there are in the rest of the sympathetic nervous system, but rather that medulla itself releases the hormones. Interestingly, as we proceed to the right-hand side in our diagram here, there are other autonomic nervous system, other hormones are controlled by the autonomic nervous system. And so in this case, we have the parasympathetic nervous system. We have our autonomic ganglion, we have our postganglionic neuron, and then we have our endocrine gland or cell. And so good examples here would be insulin and released by the pancreas, and gastrointestinal hormones, released, of course, by the GI tract. And those hormones, both of those, are released, their release is increased by stimulation from the parasympathetic division of the autonomic nervous system, and decreased by stimulation from the sympathetic division. So it's another good example of hormones whose release is controlled by neurons. Now, as we make our way further to the right here, what we notice is this. We have the third category, hormones whose release is controlled by other hormones. The most prominent example of this is in the anterior pituitary. In this particular case, we have hormones that are released in the hypothalamus. Those hormones go to the anterior pituitary, and they have a positive or negative impact. We talked about dopamine and prolactin earlier. So they have a positive or negative impact on the release of hormones from the anterior pituitary. Those hormones then go into the bloodstream and circulate through the body. So interestingly, the hypothalamus releases hormones whose only purpose is to make its way a short distance down this little portal system, as you probably recall, to the anterior pituitary and cause release of those hormones. The posterior pituitary is mentioned here, but is a little bit different. It's kind of a combination of the previous two release mechanisms we talked about. In the case of the posterior pituitary, we have two hormones that are produced in the hypothalamus by neurons, and they're transported via these neurons to the posterior pituitary and released by the posterior pituitary. So in this particular case, the posterior pituitary is not releasing hormones at all, but rather it is let me rephrase that. It's not synthesizing hormones at all, but rather it's releasing hormones that were produced up in the hypothalamus. Lastly, what we see is this, that third mechanism of hormone release, when hormones are controlled by other hormones, Well, in that case, we refer to the hormones that cause the release of other hormones as tropic hormones. These are hormones, as I stated, that are produced to stimulate the production of other hormones. Now, oftentimes, these tropic hormones also stimulate the growth of the gland that they target. For instance, thyroid stimulating hormone is produced by the anterior pituitary and goes to the thyroid gland, causing the release of thyroid hormone. And so, in this sense, thyroid stimulating hormone is a tropic hormone. On the other hand, thyroid stimulating hormone also controls the size of the thyroid gland. And so this makes it a trophic, in other words, to feed hormone. It controls the size of its target gland. And so this, of course, is a picture of a goiter. Goiters are typically the result of overstimulation of the thyroid gland by thyroid stimulating hormone for one reason or another, reasons that we'll talk about in the thyroid hormone section coming up shortly.